Hi 10th graders, hope you're doing well. We miss you. I know it's hard to receive classes this way, but it's time to stay home and to stay safe, so please stay safe. Well, today I'd like to discuss the process of cell division. For starters, the cell cycle is how the cells reproduce. All cells store genetic information in chromosomes, and most prokaryotic cells have a singular circular chromosome. This chromosome is found in the cytoplasm, as you can see in this figure. In eukaryotic cells, on the other hand, DNA is packaged into multiple chromosomes. The structure of the eukaryotic chromosomes has levels of organization within the chromosome structure, as we can see in this diagram. For example, you have the DNA double helix, as you can see there. Then you have the histone proteins and nucleosomes. These form the coils and supercoils. And then you have the centromere, sister chromatids, and duplicated chromosomes. Nucleosomes are composed of DNA wrapped around histone molecules, and these are tightly packed to form coils and coil structures. Okay, so prokaryotic cell cycle is a binary fission. So they undergo the binary fission through a different sort of steps, as you can see in this diagram. Most prokaryotic cells begin to duplicate their DNA once they have grown into a certain size. But why do they need to duplicate their DNA? Simple. The cells duplicate its DNA so that each daughter cell will have a complete copy of the original cell's DNA, so that the copy will be exactly the same as the daughter cell as the original cell. Once the DNA duplicates, the two molecules attach to different regions of the cell membrane, and the cell begins to pinch inward. If the cell membrane did not indent and pinch off, the cell would not form two new cells. The original cell would not maintain two complete copies of its DNA. This is simple. The cell cycle, even though the diagram does not draw as a cycle, it is a cycle. According to what we have been seeing, binary fission is an example of asexual reproduction. That's because two daughter cells are genetically identical to the single parent cell. Now let's talk about eukaryotic cell cycle. Eukaryotic cells have more complex cell cycle than the prokaryotic cells. Let's focus on each phase of the cell individually. First, we have the interphase. Cells spend most of their time in interphase, which is divided into three phases: G1, S1, and G2. Cells do most of their growing during G1 phase. It begins when mitosis is complete and ends when DNA replication begins. In S phase, DNA is synthesized as chromosomes and replicated. In G2 phase, many of the molecules and cell structures are required for the cell division are produced. Usually this is the shortest phase of the cell cycle. There is usually a misconception that cells get smaller with each successive cell division. However, cells grow during interphase, and the cell division helps a cell avoid the problems of growing too large, as we saw last lesson. Which cell cycle is the plant cycle? Which one do you think it's the plant cycle? There you go. That's because plants are eukaryotic, and then bacteria are prokaryotic, and the bacteria go through a binary fission. Okay, let's talk about M phase. Cell division occurs during M phase. M phase is usually much shorter than interphase. M phase results in 2 daughter cells. The first step of M phase is mitosis. The cell's nucleus divides during the mitosis. Mitosis can be divided in 4 phases that lead up to cytokinesis. The cytoplasm divides and the 2 cells are formed during cytokinesis. Multicellular organisms grow by adding more cells rather than by allowing individual cells to grow infinitely larger. Cell division helps the cell avoid problems associated with the cells that are too large. Prophase. The nucleus condenses and the chromosomes become visible. The spindle begins to form. When are the chromosomes duplicated? During S phase of the interphase. Here we can see the sister chromatids. The nuclear envelope breaks down at the very end of the prophase. Now we have metaphase. During metaphase, the centromeres of each of the duplicated chromosomes line up across the center of the cell. Spindle fibers connect the chromosomes to the 2 poles of the spindle. The 2 strands of each chromosome contain identical genetic information, as you can see here. Anaphase. During anaphase, the separated sister chromatids, now called chromosomes, move along the spindle fibers to opposite ends of the cell. Here we can see the individual chromosomes. What is the importance of this step in mitosis? What would happen if one of the duplicated chromosomes did not separate correctly during anaphase? Well, the daughter cells would not have the correct genetic information. Therefore, it would not be an exact duplicate of the original or parent cell. Telophase. In telophase, the cell begins to divide into daughter cells. Nuclear envelopes reform. The spindle begins to break apart, and the cell membrane begins to pinch together along the middle of the cell. Here you can see the nuclear envelopes reforming. And finally, we have cytokinesis. Cytokinesis is the final step in cell division. This step usually takes place during telophase. In animal cells, the cell membrane pinches in the center to form 2 daughter cells. Cytokinesis is different in plant and animal cells. In animal cells, the cell membrane is drawn inward until the cytoplasm is divided into 2 nearly equal parts. On the other hand, in plant cells, a cell plate forms near the center of the cell. This plate gradually develops into the cell membranes, and then the cell wall forms between 2 daughter cells. You can see this in your textbook. The chromosomes of 2 daughter cells are genetically identical. Mitosis is a form of asexual reproduction then, because you have 2 daughter cells that are identical. Okay, let's go through the mitosis overview. First, although interphase and cytokinesis are shown in this diagram, these are not part of mitosis. Let's just be clear about that. During interphase, a cell must grow and replicate its DNA for it to be able to divide. Okay, so we're going to see each part of it. Its stage. Prophase. Here, chromatin condenses into chromosomes. The centrioles separate, the spindle begins to form, and the nuclear envelope breaks down. In metaphase, the chromosomes line up across the center of the cell. In anaphase, the sister chromatids separate and move along the spindle fibers toward the opposite ends of the cell. In telophase, the chromosomes gather at the opposite end of the cell and lose their distinct shapes. Two nuclear envelopes form. Finally, we have cytokinesis, which is the final step of cell division, bringing about the separation of the two daughter cells. And that's it, guys. Thank you for your attention.